Okay, this video is going to be covering ketogenesis. This is the synthesis of what are considered ketone bodies. Now, ketogenesis occurs in the fasting state in the liver. The liver is responsible for making ketone bodies. So we are going to be focusing on the liver and a fasting state. Now, in order to orient ourselves with the response of ketogenesis in a fasting state, let's look at our time course graph that we've seen before. But first, notice on the x-axis we are looking at days of fasting for ketogenesis. Recall when we talk about the initial fed and fasting states, we're talking about hours. So on this scale here, we're talking approximately 30 to 40 days. Now, the main feature of ketogenesis is that you can consider it a last-ditch response of the body taking care of itself before starving to death. And the purpose of ketogenesis is largely to produce energy for the whole body, but it's mainly to supply the brain with sufficient fuel in declining glucose conditions. So now if we look at glucose, we'll see that, remember, when we're getting out into days here, in the matter of 2 to 4 days, the amount of glucose that is being supplied by glycogenolysis is almost exhausted, is essentially completely exhausted here. So the glucose that's being supplied into the bloodstream is coming from, at this point, gluconeogenesis. And again, this is dependent on the supply of precursors, of substrates for gluconeogenesis from skeletal muscle, other cells of the body, and adipose. So the glucose being supplied to the blood is, after a couple of days, it's going to decline. And this decline takes blood glucose from a condition of normal homeostatic levels, and it brings the glucose down to subnormal levels. Now, these subnormal levels are essentially at the edge of losing consciousness, and the drop in glucose production is called glucose sparing. And the reason for glucose sparing is because, remember, the substrates that are going into the gluconeogenic process, we are capitalizing on a large amount of amino acids, on lactate, glycerol, and the glucose sparing response is the body's way of basically getting off the throttle of the liver for the body shifting into what it is considering a long-term fast or starvation state. Now, the drop in glucose from normal to subnormal levels is insufficient for normal brain function, metabolic function. So there has to be some other fuel source that is taking up this slack. Now, also recall that we do have, because we have glucagon signaling, we have a sustained duration of fatty acid release from adipose that will undergo beta oxidation reactions. Now, the free fatty acids, of course, can be taken up by most cells, except the brain, and used as an alternative fuel. This is also, of course, presupposing that there is a supply of adipose tissue in order to release fatty acids. Once we start getting out into long-term starvation states, 30 to 40 days, now we start running into other physiological problems. But as long as we have adipose tissue, we can have fatty acid release, which can supply some much-needed energy substrates for especially skeletal muscle. Now, again, the brain undergoes essentially, consider no oxidation of fatty acids, no beta oxidation. So the drop in glucose concentration in the blood needs to be made up by something. And the first thing that we notice that is being synthesized over long periods of time here is another fuel source that is called acetoacetate. Acetoacetate is a ketone body, and it's one of the two ketone bodies we're going to be looking at. Another ketone body, we can take a look at it as this line. Obviously, this is a much more substantial ketone body that's being synthesized, and this one is called beta-hydroxybutyrate. So these are the two ketone bodies that we are going to be concerning ourselves with. The first is acetoacetate. The second is beta-hydroxybutyrate. And these two ketone bodies are essentially the fuel sources that make up for the glucose sparing response by the body and the drop in gluconeogenesis. The brain can very readily use both of these fuel supplies, as can most other cells, especially, of course, skeletal muscle. So notice also here the amount of ketone bodies synthesized. The beta-hydroxybutyrate is synthesized at a much greater concentration than the acetoacetate is. And we'll get into this when we look at the reaction schemes of how glucose or how ketone bodies are synthesized. Okay, next slide, we need to review the essential overview of the ketone body metabolism and take a look at all of the responses in general, how the liver is coordinating with the rest of the body. So, again, ketone bodies are synthesized in the liver. The source of ketone bodies is acetyl-CoA. The acetyl-CoA by the liver, we'll take a look at these reactions in a minute, will combine to form acetoacetate. These then will used to form beta-hydroxybutyrate. Once the beta-hydroxybutyrate is formed, as well as the acetoacetate, both of these can exit the liver and enter the bloodstream. And these are our ketone bodies. These ketones, now that they are in the blood, they can go to primarily brain and, of course, our friend skeletal muscle, which will use any fuel sources available. Then these are broken back down into acetyl-CoA, and then these can be used for energy generation. Now, the question is in the liver, we need a substrate. We need a source of acetyl-CoA. So what is the source of acetyl-CoA? Well, remember, we are in a fasting, essentially starvation state. So that means we have substantial glucagon signaling. So under fasting conditions with glucagon signaling, what is a major energy substrate for liver? Well, glucagon, remember, causes fatty acid release from adipose tissue. These fatty acids enter the liver. The liver undergoes beta oxidation in the mitochondria, and generates acetyl-CoA. And remember, enormous amounts of acetyl-CoA. Now, the energy state is very high. Okay, so pardon the quacking of my phone. That was my better half asking if we wanted to plant some grapes in the back, and it's a go. Okay, so back where we were. Fasting state, starvation, glucagon signaling, fatty acid release, adipose releasing the fatty acids, liver taking up the fatty acids. Fatty acids undergo beta oxidation, generating large amounts of acetyl-CoA in the liver. Now recall that this acetyl-CoA is not going to be used to be making fatty acids because we are in a fasting glucagon signaling state. But the acetyl-CoA is going to build up inside the mitochondria. And as this acetyl-CoA starts to build up in the mitochondria, it essentially has no place to go. Remember that this is inhibiting pyruvate dehydrogenase, and the acetyl-CoA is activating pyruvate carboxylase, so that this is going to push this direction into gluconeogenesis. Now, the acetyl-CoA is not going to be condensing. Once it's formed, this high levels of acetyl-CoA, it is not going to be condensing with oxaloacetate to make citrate. And the reason for this is because, think about where the oxaloacetate is going. The pyruvate carboxylase is transforming pyruvate into oxaloacetate, which is becoming malate because of the very high levels of NADH in the mitochondria. And then the malate is exported out into the cytoplasm for gluconeogenesis. So essentially, once the oxaloacetate is formed, it's immediately transformed into malate, which is disappearing rapidly from the mitochondria, which is going to push the malate dehydrogenase enzyme into the reduction way to reduce oxaloacetate into malate. Therefore, the concentration of oxaloacetate is going to be near zero under these states, and so there is none available for the condensation reaction with acetyl-CoA. Because the condensation into citrate is blocked, really the only way that acetyl-CoA is going to be transformed is into ketones. Okay, so now let's look at the few reactions that are required for ketone synthesis. So again, we are going to start out with our acetyl-CoA. And in fact, we are going to start out with 2 acetyl-CoAs for the first step of ketone synthesis. This is an enzyme that you are familiar with. This is the thiolase enzyme. And we are going to be removing a CoAsh on this, and we are going to form something you've seen before. This structure is acetyl-acetyl-CoA. Now recall where you've seen this before with the thiolase reaction. This has happened as the first step of cholesterol synthesis in the cytoplasm. But keep in mind, this is acetyl-CoA in the mitochondria. So this is the mitochondrial form of thiolase that mediates this reaction. Now the next reaction is another one that you have seen before. And this reaction is mediated by HMG-CoA synthase. And here we are forming HMG-CoA. Sound familiar? So this is the second step of cholesterol synthesis. But again, keep in mind, this is the mitochondrial enzyme that is responsible for converting the acetoacetyl-CoA into HMG-CoA. And of course, in this reaction, we are losing another CoA. Okay. So now, next step. We have our HMG-CoA structure, which I will draw out for you here. And the next reaction here is going to convert this structure into acetoacetate. So this step is converting HMG-CoA into acetoacetate, and we are losing, in this reaction, an acetyl-CoA group, which is right here. So by forming this reaction in the mitochondria, we are now taking advantage largely of the very, very high levels of acetyl-CoA and constructing and driving the synthesis of acetoacetate. So this is our first ketone body. And this last step is mediated by HMG CoA lyase. Now recall, this step here in the cytoplasm is mediated by HMG CoA reductase to form mevalonate for cholesterol synthesis, but again, that's the cytosol. This is the mitochondria that has HMG CoA lyase, and it mediates the removal of this acetyl-CoA off of the HMG CoA structure, giving us our acetoacetate. Now the next step, this acetoacetate can be secreted by the cell into the blood and supply the body with a ketone for fuel. But let's take advantage of this particular carbon right here. You'll notice that this carbon is in an oxidized state. So perhaps we could take this and perform a redox reaction and load a little extra energy onto this acetoacetate structure, like so. And in doing so, we can oxidize our NADH in the mitochondria which, remember, the concentration of NADH in the mitochondria in this fasting starvation state in the liver is very, very high because we have so much beta oxidation going on. And so by taking this molecule of acetoacetate, which is essentially two acetyl-CoA groups, and putting it out into the blood, we're not taking advantage of the possibility of reducing this carbon. So if we take advantage of this and reduce this carbon by beta-hydroxy butyrate dehydrogenase, using all of that rich NADH in the mitochondrial matrix, we can reduce this carbon and put an alcohol group on there. And so this becomes beta-hydroxy butyrate. Butyrate because we have 4 carbons. And remember your nomenclature. This is the alpha carbon, this is the beta carbon. Looks like a fatty acid. So this is the beta-hydroxy group. So beta-hydroxy butyrate. Now the beta-hydroxy butyrate, once it's formed, of course, can be secreted by the liver into the blood. Now we have these two structures, beta-hydroxy butyrate and acetoacetate. These two represent our two ketone bodies, of which, of course, the beta-hydroxy butyrate is of higher concentration because we have this oxidized carbon on the acetoacetate that we can load up a little extra energy in the form of electrons. So we are going to take this carbon and reduce it to its reduced form here with the hydroxyl group on the beta carbon. Now both of these in the blood. Now one reaction that can also happen is the acetoacetate can spontaneously transform in the blood, and this is a non-enzymatic mediated reaction. And how it can spontaneously form, what it can spontaneously form is this. Acetone. And by doing so, the acetone will be cleared out by the lungs. Now, I think most people have smelled acetone. It's kind of that sweet smell. And so this is the reason why, especially in alcoholics that are in a fasting state, because alcoholics tend not to have very good nutrition, so they're not eating well, but they are drinking large amounts of alcohol, they produce these ketone bodies. And these ketone bodies in their bloodstream are acids because, remember, these groups have carboxylic acid groups. So the higher the concentration of these ketone bodies in the bloodstream, the higher the concentration of acid in the blood, which decreases the pH. This obviously can cause serious problems with enzymatic reactions. And the higher the concentration of acetoacetate, the higher the spontaneous reactions are going to occur for acetone production, which is going to be expired by the lungs, hence that sweet smell. So those of you that are interested in clinical aspects of it, when an individual that's an alcoholic under poor nutrition comes into the ER and they smell of acetone, they are almost invariably in a ketoacidotic state. And the ketoacidotic state is reflected by the carboxylic acids that are being put into the blood at high concentrations, which will drop the pH. Now, is this bad that we have ketone bodies? No, because this is a major fuel source for the brain and skeletal muscle during extended starvation states. Can we live in this ketoacidotic state forever? Of course not, because as long as there's fat available, the liver will continue making ketone bodies until the individual can reverse the state by introducing a fuel source. Now, the last step of talking about ketone metabolism is breaking down the ketones once they get into the tissue that is going to be using these ketones. So let's start out with beta-hydroxybutyrate. This is taken up. Let's stick with our important organ, the brain. So this will go into the brain. We'll use this as an example, but remember, skeletal muscle can do this. Many cells can do this response. The first step is that the cell is going to capitalize on that little bit of extra energy that was brought along by the beta-hydroxybutyrate by performing a redox reaction on the the acetoacetate. So we are going to reverse that reaction, taking our beta-hydroxybutyrate, oxidizing it into acetoacetate. And this is simply the beta-hydroxybutyrate dehydrogenase enzyme in reverse. So these reactions are also occurring in the mitochondria. So the next step of this reaction is to take our acetoacetate and process this group by charging it up, as we have seen before, because we need to prepare the acetoacetate for subsequent reactions. And we are going to charge this up with acetyl-CoA. So again, it's the reverse of these reactions. So this is the acetyl-acetyl-CoA. This enzyme is going to require a donor of the acetyl-CoA, and the donor here is going to be succinyl-CoA. Will donate its CoA factor, generating succinyl and producing that high energy bond with the acetoacetyl-CoA. Then finally, the next reaction is mediated, as you can guess, the reverse, which is the thiolase reaction. And this is going to generate two acetyl-CoAs. And here we are introducing a CoA, coenzyme A, into the reaction so that this coenzyme A can attack this carbonyl and form our two acetyl-CoAs. And now the two acetyl-CoAs obviously go into the TCA cycle, because we're in the mitochondria, of the brain cell or of skeletal muscle, and generate our much needed ATP.